Thank you very much. This is going to be a quick presentation just highlighting some of the applications of the various tools that have been developed. In this instance, we're looking at the human epidermal growth factor receptor 2, ordinarily expressed at low levels. But in the case of HER2 enriched, the HER2 enriched molecular subtype, there's expanded expression. This occurs in up to 30% of breast cancer cases. So on the one hand, there are the hormone receptors, ordinarily cytosolic, until they're activated, and then there are the epidermal or the growth factor receptors, which tend to be in the plasma membrane and get bound by the cognate growth factors. So here we have here the molecular subtypes as we know them: luminal A, and luminal B, which are hormone receptor positive, except that in the case of luminal B, there's also the human epidermal growth factor. In contrast to the luminal A, which doesn't have the HER2 expression. Intervention therapeutically involves the monoclonal antibodies, which are rather specific. And trastuzumab has been in use for a while. There are newer drugs working by the same method, typically binding to the HER2 receptor. There's ubiquitin ligase activity, there's tagging, and then there's breakdown of the human epidermal growth factor receptor. So by that means the drug is used for therapy and has made a difference in many cases. So the good news is that some 35% of cases will respond to these monoclonal antibodies. In the case of trastuzumab, of those who respond, about 70% will have the resurgence of the disease, metastasis within one year or so. So trastuzumab resistance has been a problem. And here it is known that SAC activation, in those instances where there's trastuzumab resistance, there's a lot of activation of the SAC kinase. The PTEN phosphatase activity is also reduced. There are other factors. There's single nucleotide polymorphism that's associated with that, along with microRNAs expression pattern that's also associated with MET, which is another tyrosine kinase. So what did we do here? In our quest for some insights as to what regulates HER2, so we turned to the Cancer Genome Atlas and got some RNA-seq data from there. We looked at a whole range of samples. We used the RNA-seq by expression maximization algorithm to do our normalization. We then got our compendium of expression data, genes in rows, samples, and columns, matrix. Rather than use all 20,500 genes, we turned to the gene ontology to get a subset that is more relevant to cancer. So genes associated with cancer, apoptosis, senescence, cell cycle, oxidative stress, and signal transduction were selected. We have about 6,000 of them. And then we used the PM50 method to identify the molecular subtypes. Having identified the molecular subtypes, we had a compendium for each molecular subtype and then applied a few transcriptional regulatory inference algorithms. We use the Arachne algorithm for reconstruction of accurate cellular networks, and we use the trees-based method, GENI3. So I just want to highlight the insights we drew from some of the approaches. There wasn't unanimity between the approaches, especially if you looked only at the direct connections to the HER2 receptor. Here at the node, we see it as ERBB2. If we looked at the across all molecular subtypes, no, I'm sorry, I just. Thank you. All right, so looking at those transcriptional regulatory algorithms, we'll look here across all the molecular subtypes where we used the entire compendium. We find that there's more constraint on the regulation of the HER2 gene, so that we have, for instance, the hormone receptor, progesterone receptor, directly constraining the expression of B2. We've found that also in the luminal B, which also has both hormone receptors along with the growth factor receptor. We find here in that instance that the estrogen receptor 2 regulates the expression of that. So there's a lot more constraint on the B2, where we don't have the crosstalk. Ordinarily, there's crosstalk between the hormone receptors and the growth factor receptors. That is missing in HER2-enriched HER2-enriched scenario because there are no hormone receptors to speak of. So there's less constraint. That's one of the observations we make. And then we find that there are essentially three regulators. Regarding ERF2, it is already known that it regulates various aspects of the expression of B2, so that's previously known. What is novel here has to do with the homeobox5, HOXA5 gene, which is a transcriptional regulator. Its loss is one of the hallmarks of aggressive cancer, and we find that it regulates the B2 along with the circadian cryptochrome 2, which also, when it has decreased expressions associated with the high-grade aggressive cancer. We find that those two directly constrain the expression of B2. This is just a number of the molecular insights gleaned from applying the inference algorithms, and I thank you for your attention.
Okay, and our last talk before the break is Alice Yu, and she's going to be talking about a network-based approach to identifying cell-cell interactions within the lung tumor microenvironment. And Alice is from Stanford University.